Biochemistry 401, Lecture 20. Today we're going to talk about the TCA cycle. This is a cycle that involves 8 enzymes, and its purpose is the oxidation of fuel and the formation of anabolic precursors. This cycle also produces 1 GTP, 3 NADH, and 1 FADH2. We're going to discuss key reactions of this cycle and its regulation. These notes are color-coded to make things easier for you. The enzymes are colored in blue and are italicized. Inhibitors are red, positive effectors are green, and important anabolic products are pink. Some key thoughts regarding regulation are shown below. There are primarily 3 modes of regulation in the cycle. The first is the availability of substrate. As metabolites build up in the cell, this increase in substrate will drive reactions forward. When these substrates are scarce, the reactions will slow down. Some enzymes are also inhibited by their products.
And finally, there is negative feedback by downstream intermediates.
This is the TCA cycle. TCA stands for tricarboxylic acid. This is another name for citrate, which appears at the top of the cycle. If this were a clock, citrate would be at 12 o'clock.
Citrate's 3 carboxylic acid groups are highlighted in red.
This cycle is also called the citric acid cycle and the Krebs cycle.
For Hans Krebs, who elucidated much of this cycle.
Now, in looking at this, it is important that you know the names of the enzymes and the metabolites that they form. It is also important that you know where NADH, FADH2, carbon dioxide, and GTP are made. As we go further on into this lecture, you're going to see that many of the intermediates in this cycle can be used for other things besides just the production of energy. And it's important that you know those as well. One thing that I'd like to tell you is that in the next exam, you will not be asked to discriminate between real names and fake names. If you see an enzyme name on the exam, it will be an actual enzyme, so you're not going to have to distinguish between isocitrate dehydrogenase, for instance, and isocitrate synthetase or something like that. Please note that there are 2 enzymes in this cycle that sound pretty similar: succinyl-CoA synthetase and succinate dehydrogenase. Now, the citric acid cycle is the hub of metabolism. This is where fuels go to be oxidized. A lot of the intermediates are also very important for other pathways. Alpha-ketoglutarate and oxaloacetate are important both for the synthesis of nucleotides and amino acids. Succinyl-CoA is important in the production of porphyrins, and so truly, the TCA cycle lies at the center of metabolism.
So let's look at this as an overview to really figure out what's going on here. We're going to start and end with a 4-carbon molecule, oxaloacetate. We're going to bring in 2 carbons in the form of an acetyl group to yield a 6-carbon molecule. Then there are going to be 2 oxidative decarboxylations in succession. Each one will yield NADH and CO2. After the first one, we'll have 5 carbons. After the second one, we'll have 4 carbons. Following this, we're going to form GTP or ATP, depending on the tissue, FADH2, and the last NADH to end up with oxaloacetate. And so in this process, there are 3 molecules of NADH that are produced, 2 molecules of carbon dioxide, a molecule of either GTP or ATP, depending on the tissue, and 1 molecule of FADH2, and this is for every complete turn of the cycle, going from oxaloacetate at the start and ending at oxaloacetate. Let's look at this a little more closely before we go into the individual reactions. And so oxaloacetate is going to combine with 2 carbons from acetyl-CoA. This is going to occur through a condensation reaction, which produces the 6-carbon molecule citrate. Then citrate is going to undergo an isomerization. Water is going to leave, and then water is going to come back again to form isocitrate. And again, this is an isomerization reaction that involves a dehydration and rehydration. The next thing that's going to happen is the first oxidative decarboxylation to produce alpha-ketoglutarate. And since this is a redox reaction, we have to have an electron carrier. The electron carrier is NAD+. NAD+ accepts electrons 2 at a time as a hydride ion, and the extra proton is released into solution. And so at the end of this reaction, we have 5-carbon alpha-ketoglutarate, carbon dioxide, and NADH plus a proton. The next thing that's going to happen is we're going to have a second oxidative decarboxylation. This happens in reactions that are a lot like what happens in the pyruvate dehydrogenase complex. Very similar enzyme complex, conducting very similar reactions. And what we're left with at the end is succinyl-CoA, carbon dioxide, NADH, and a proton. Again, this is a second oxidative decarboxylation, and we've now produced 2 molecules of NADH. We've released 2 molecules of carbon dioxide, and now we're left with a 4-carbon intermediate, succinyl-CoA. Now this is an activated intermediate. The next thing that's going to happen is pretty big. We're going to make ATP, and this occurs through a substrate-level phosphorylation. And so we're going to use GDP or ADP, it depends on the tissue which one is used, and inorganic phosphate to make GTP or ATP. And what we're left with is succinate. This is a 4-carbon molecule, and we've produced also, of course, the GTP or the ATP, and coenzyme A is also released. And again, this happens through substrate-level phosphorylation. The next thing that's going to happen is we're going to take this succinate and we're going to get all the energy that we can out of this. So we're going to make sure that this is oxidized as much as it can be. The next thing that's going to happen is we're going to, in fact, oxidize this with FAD as the electron carrier. And we're going to end up with FADH2 and fumarate, which is a 4-carbon molecule. Now the next thing we're going to do is we're going to hydrate this fumarate. We're going to add water in order to form malate. Now we've seen malate before, and in order to get from malate to the starting material oxaloacetate so we can start all over again, we're going to oxidize malate to form oxaloacetate. And the electron carrier is going to be NAD+, and we're going to end up with NADH and a proton and oxaloacetate, the 4-carbon molecule. And so now we're back at the beginning. And the reducing equivalents, the NADH or the FADH2, are going to be used in the electron transport chain in order to make ATP.
Here we see reaction 1. In this reaction, oxaloacetate, which contains 4 carbons, is going to condense with acetyl-CoA that has 2 carbons to form citral-CoA, which is the intermediate. This intermediate is going to lose the coenzyme A through hydrolysis to yield citrate and coenzyme A. Citrate is a 6-carbon molecule. Again, this is a condensation reaction, and the product is a 6-carbon molecule, citrate. The enzyme that catalyzes reaction 1, citrate synthase, is a dimer. This enzyme operates by sequential ordered reaction. Oxaloacetate binds first and causes a small subunit of citrate synthase to rotate to form the acetyl-CoA binding pocket. This also brings closer those residues that are necessary for the condensation reaction between oxaloacetate and acetyl-CoA to occur. And so in this way, the enzyme is only active when both substrates are available. This is a prime example of an enzyme that works by induced fit.
The regulation of this enzyme occurs through inhibition via ATP, NADH, and succinyl-CoA, and these are all an example of negative feedback regulation. And so now on to reaction number 2. This is catalyzed by the enzyme aconitase and consists of an isomerization that occurs in two steps. The first step is a dehydration of citrate to yield a double bond between the central carbons. This results in the intermediate cis-aconitate. The second step is a stereospecific rehydration of that double bond to yield isocitrate. The water is going to be added in a specific manner so that we go from a tertiary alcohol to a secondary alcohol. One thing I would like to point out is that citrate has a plane of symmetry at the central carbon. If you'll notice, there is a carboxylic acid and then a methylene group, CH2, and then the central carbon in the upper portion and the lower portion of this molecule. And so this molecule has symmetry. But that is not the case with isocitrate. This molecule has a carboxylic acid and then a secondary alcohol. And so with this dehydration and rehydration, we've introduced an asymmetry in the molecule, and this is going to set up for the oxidative decarboxylation in the next step.
In reaction number 3, the enzyme isocitrate dehydrogenase is going to catalyze an oxidation and a decarboxylation reaction. The first thing that's going to occur is an oxidation-reduction reaction. Isocitrate is going to be oxidized and NAD+ is going to be reduced. This produces our first NADH. This forms an unstable beta-keto acid intermediate, oxalosuccinate, which will rapidly become decarboxylated to form alpha-ketoglutarate and carbon dioxide. And so this is the first reaction in which carbon dioxide is produced and in which NADH is also produced. This NADH is a reducing equivalent that will be used to make ATP.
This enzyme catalyzes the rate-limiting step and is regulated in a positive manner by ADP and NAD+, which indicate a low energy charge in the cell. Negative regulation is through NADH, which directly competes with NAD+ for the active site, and through ATP, which is an allosteric regulator.
The fourth enzyme is actually an enzyme complex, the alpha-ketoglutarate dehydrogenase complex. This is similar to pyruvate dehydrogenase complex. The reaction that alpha-ketoglutarate dehydrogenase complex catalyzes is shown below, in which alpha-ketoglutarate plus NAD+ plus coenzyme A yields succinyl-CoA plus carbon dioxide and NADH. This reaction results in addition of coenzyme A and release of carbon dioxide, and the second molecule of NADH is produced. And we end up with a high-energy intermediate, succinyl-CoA. Now again, whenever you see coenzyme A, please note that the next reaction is going to be a big one. We've loaded this molecule up with coenzyme A to do something relatively big, to make GTP in the next reaction. The enzymes that are involved in alpha-ketoglutarate dehydrogenase complex are similar to those that are seen in the pyruvate dehydrogenase complex. In fact, E1 and E2 are homologous to those that are seen in PDC. And E3 is identical to PDC E3. Together, these three enzymes make up the alpha-ketoglutarate dehydrogenase complex, and it acts in much the same way as pyruvate dehydrogenase acts. We're going to add coenzyme A, and we're going to liberate carbon dioxide and yield a reducing equivalent, NADH. This reaction is driven forward through the loss of carbon dioxide.
The alpha-ketoglutarate dehydrogenase complex is also a major site of regulation. This regulation is through AMP, which positively regulates the enzyme, and through product inhibition by succinyl-CoA and NADH, which both downregulate the enzyme.
In reaction number 5, succinyl-CoA, GDP, and inorganic phosphate are going to yield succinate plus coenzyme A and GTP. This is a substrate-level phosphorylation of GDP to yield GTP, and in the process, coenzyme A is released. The enzyme that catalyzes this reaction is succinyl-CoA synthetase. Synthetases are enzymes that require a nucleotide triphosphate, in this case GTP. Succinyl-CoA synthetase is actually named for the reverse reaction, in which succinate and GTP and coenzyme A yield GDP inorganic phosphate and succinyl-CoA.
In reactions 6 through 8, we're going to oxidize the 4-carbon molecule succinate and end up with oxaloacetate through a series of 3 reactions. The first one is an oxidation-reduction reaction, in which FAD is used as an electron carrier. The second step, reaction number 7, is a stereospecific hydration across the double bond to yield malate. And the last step, reaction number 8, is another oxidation. We're going to use NAD+ as the electron carrier. And so what we have here is an oxidation, hydration, oxidation. And this is the best way to get as much energy out of this 4-carbon molecule as we can. Now we're going to see the same progression of reactions in the oxidation of fatty acids a little bit later on in the semester. Oxidation with FAD, hydration across the double bond that results, and then an oxidation using NAD+ as the final electron carrier. And in the TCA cycle, we're going to end up where we started, with oxaloacetate. Let's look at each of these reactions one at a time.
This is reaction number 6. The enzyme that catalyzes this reaction is succinate dehydrogenase. It catalyzes the oxidation of succinate to form fumarate, with the reduction of FAD to form FADH2. We're going to use covalently attached FAD as a cofactor. The electrons from succinate are transferred to iron-sulfur clusters within the succinate dehydrogenase enzyme. Ultimately, oxygen will accept these electrons by way of the electron transport chain. This is because succinate dehydrogenase actually is involved in the TCA cycle and in the electron transport chain, and we'll look at this a little bit later on. In this reaction, FADH2 is a product, and this enzyme is regulated by the FADH2-FAD ratio. High ratios of FADH2 will inhibit this enzyme. The flavin ring in succinate dehydrogenase is covalently attached to the enzyme itself, and succinate dehydrogenase is embedded in the inner mitochondrial membrane. This allows succinate dehydrogenase to function both in the TCA cycle and in the electron transport chain. In reaction number 7, we're going to hydrate fumarate to form malate. We're going to add water across the double bond to set up for the final oxidation of the TCA cycle, and the product is malate. We saw this intermediate when we were looking at gluconeogenesis. Malate is an important product, as it can also be used in gluconeogenesis.
Reaction 8 is the last reaction, and it's catalyzed by the enzyme malate dehydrogenase. In this reaction, malate is oxidized and NAD+ is reduced. And the products are NADH plus a proton and oxaloacetate.
And so with reaction 8, we're back at the starting point. We've regenerated oxaloacetate to restart the cycle. This enzyme is downregulated by a high NADH to NAD+ ratio. Now this reaction is unusual in that it has a standard free energy change of 7 kilocalories per mole, and we drive this reaction forward by removing the product, oxaloacetate. And so let's think about this for a minute. Oxaloacetate can be removed by making citrate. When there are sufficient quantities of acetyl-CoA and oxaloacetate will condense to form citrate, and so the molar concentrations of oxaloacetate will decrease, and this will drive reaction 8 forward. If we need oxaloacetate to make either nucleic acids or amino acids, this will also decrease the molar concentration of oxaloacetate as well. And this in turn will drive reaction 8 forward. But when there's a lot of oxaloacetate around, this indicates that the TCA cycle for some reason has slowed down. And so it's a good idea to reverse direction and make malate to shuttle it out of the mitochondrion to make glucose.
The 4 major points of regulation of the TCA cycle are shown here. These are the pyruvate dehydrogenase complex, citrate synthase, isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase complex. The pyruvate dehydrogenase complex is upregulated by high levels of its substrates, NAD+ and coenzyme A. It is downregulated by high concentrations of its products, acetyl-CoA, NADH, and ATP. Citrate synthase is downregulated by succinyl-CoA, NADH, and ATP. These are all downstream products of the TCA cycle that feed back to downregulate the activity of citrate synthase and thereby modulate the entry of acetyl-CoA into the cycle. Isocitrate dehydrogenase catalyzes the rate-limiting step for the TCA cycle. It is upregulated by NAD+ and ADP and is downregulated by NADH and ATP. Alpha-ketoglutarate dehydrogenase complex is upregulated by AMP and is downregulated by succinyl-CoA and NADH. In addition, individual reactions can be regulated at the level of their products and reactants by Le Chatelier's principle.
Many of these products that are made in the TCA cycle are used for other things. Citrate is used to make fatty acids and also sterols, such as cholesterol and the steroid hormones. Alpha-ketoglutarate is used to make glutamate, other amino acids, and also nucleotides. Succinyl-CoA is used to make porphyrins that are necessary for heme and chlorophyll. Oxaloacetate makes the amino acids and nucleotides. Now the thing is, if we're going to use all of these intermediates to make other metabolites, we run a danger of depleting the cycle. And so there are reactions to fill up the cycle again so that we do not deplete these intermediates. Reactions that synthesize these intermediates are called anaplerotic reactions. This means filling up in Greek.
The major anaplerotic reactions that fill the TCA cycle are shown here. You're only responsible for the one in yellow because you've seen this one before. Do you remember where? That's right, gluconeogenesis. Good for you. We'll see some of these other reactions later on in the semester, but for now, this is the one that you need to know.
So what would happen to the TCA cycle if succinyl-CoA were continually removed to make heme for hemoglobin, and the only input into the cycle was acetyl-CoA? Would the rate of carbon flow through the cycle accelerate? Would it slow and eventually stop? Or would there be no change at all to the cycle? If we were to remove succinyl-CoA for the production of porphyrins, we would soon run out of oxaloacetate. We need oxaloacetate to replenish the cycle. And so if we were constantly to remove intermediates, we would eventually slow the citric acid cycle. Now, mammals cannot use acetyl-CoA for net conversion into oxaloacetate. We lack the enzymes necessary for this to occur. Acetyl-CoA must enter the TCA cycle by way of condensation with oxaloacetate to form citrate, and there is a loss of the 2 carbon dioxides for every acetyl entry into the cycle. We cannot use acetyl-CoA for this.
For every acetyl-CoA that comes in, we lose 2 molecules of carbon dioxide, and so there is no net carbon gain from entry of acetyl-CoA. And so for this reason, we cannot use acetyl-CoA to make glucose. Other organisms can use acetyl-CoA as a substrate for gluconeogenesis. However, mammals cannot. We'll talk about this pathway later on when we talk about the breakdown of fatty acids, later on in the semester. This is an interesting photo by an artist whose name appears here. You should check out his other works. They're really very interesting. So now we're going to talk about this cycle in big picture terms. I hope this overview gives you an idea of how to look at metabolism. When you look at a pathway, see which reactions are irreversible and see what sorts of metabolites will be built up if those particular reactions are inhibited. Those will usually be activators or inhibitors of the pathway. Also, it's important to know whether your particular pathway is energy producing or energy using. If the cell needs energy, energy producing pathways are going to be activated and energy using pathways will be inhibited. Vice versa, if the cell has a lot of energy, let's not waste carbon metabolites for energy. If you have enough energy in the cell, it will tend to store carbon fuels. It's time to store them or to use them for synthesis. Let's not break them down. If we look at this table, we can see the delta G naught prime and the delta G for these reactions in the TCA cycle. 4 of them have negative delta Gs in the cell. These are pyruvate dehydrogenase complex, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. Those reactions that have a very high negative delta G are going to be irreversible in the cell, whereas those that are close to equilibrium will be fully reversible. So even though this figure shows that these reactions are all only going one way, for some of these, it's not true. This is actually a much more useful figure, and it comes from the book that I'm writing. I ask that you please do not disseminate this online, as it is copyrighted material and belongs to me. Thank you. From this figure, we can see that 3 of the reactions in the cycle itself are irreversible. These irreversible reactions are indicated by a blue color, whereas those that are reversible are shown in beige. In addition to these 3 irreversible reactions shown here, there is another one that lies outside the cycle, and that's the pyruvate dehydrogenase complex. This enzyme complex regulates the entry of metabolites into the TCA cycle through production of acetyl-CoA. And so the position of these 4 irreversible reactions are shown here, 1 outside and 3 inside the cycle. An easy way to think about the regulation of this cycle is to realize that it's used for energy primarily, and so it's going to be regulated in terms of high energy or low energy in the cell. In low energy state, there'll be low ratios of NADH to NAD+, ATP to ADP or AMP if we're in really bad shape, and also low ratios of acetyl-CoA to coenzyme A. These all indicate that the cell has low energy stores, and it's time to get the cycle going. In contrast, in a high energy state, these ratios will all be high, indicating that the cell has enough energy and it's time to turn this cycle down. And so these 4 enzymes in high energy states will be turned down, leading to a buildup of metabolites. This is a metabolic traffic jam, inhibition of traffic by ELK. It causes everybody to back up and some to turn around. And this is the traffic jam. And oh my, well, that's not an ELK. That's not one either. But inhibition in the TCA cycle is similar to this. You have a traffic backup that's caused by the inhibition of these enzymes. So metabolites will tend to build up. Which ones build up? That's the key. So do you remember the main regulatory enzyme in the TCA cycle? It's isocitrate dehydrogenase. This is the rate keeper for the cycle. It is the main decision point for turning up the cycle or turning down the cycle. Let's see how this happens. We said that when there's high energy state in the cell, when there's a high relative concentration of NADH to NAD+, and ATP to ADP or AMP, that isocitrate dehydrogenase will be inhibited. So what happens when the activity of this enzyme is decreased? Isocitrate will tend to build up, and then it will cause the reversible reactions to go backwards. What this means is aconitase will go backwards in the reverse direction, and once it hits citrate synthase, that enzyme does not go backwards. That is not a reversible reaction, and so citrate will tend to build up. High concentrations of citrate
are a very potent signal that the cell has enough energy and enough building blocks. And so this metabolite is used to regulate the activity of enzymes. Citrate inhibits its own production through the inhibition of citrate synthase. This is a good example of negative feedback regulation and product inhibition. Because the rate of citrate synthase has decreased, acetyl-CoA is in a traffic jam, and it will tend to build up. This buildup of acetyl-CoA, in turn, affects other enzymes. High concentrations of acetyl-CoA will inhibit the pyruvate dehydrogenase complex, and again, this is negative feedback regulation and product inhibition. And so the use of pyruvate to make acetyl-CoA is going to decline. But what do we do with this pyruvate? Lucky for us, acetyl-CoA also increases the rate of pyruvate carboxylase. And we saw this enzyme before. This is a gluconeogenic enzyme that takes pyruvate and turns it into oxaloacetate through the addition of carbon from bicarbonate and an energy source, ATP. And so now we have oxaloacetate. This buildup of oxaloacetate tends to reverse the malate dehydrogenase enzyme, and malate dehydrogenase reduces oxaloacetate to make malate. This malate can be exported into the cytosol to continue on in gluconeogenesis to make glucose. Why? Well, we can store this for later as glycogen. And so what have we done? The cell has switched gears, switching from breaking down fuels to gain energy in times of energy need, to storing fuels in times of energy abundance. And we did this by turning isocitrate dehydrogenase down, causing the increase of mitochondrial citrate. But when citrate builds up in the mitochondrion, it will also be transported into the cytosol, where it can have an effect on enzymes in the cytosol, such as those involved in gluconeogenesis, glycolysis, and fatty acid synthesis. Let's take a look at that. You will recall that citrate is a potent reciprocal regulator of gluconeogenesis and glycolysis. It inhibits the glycolytic enzyme PFK1 and the regulatory enzyme PFK2. It also activates fructose 1,6-bisphosphatase, which functions in gluconeogenesis. And so in this way, it turns down glycolysis and turns up gluconeogenesis. Citrate also increases the rate of fatty acid synthesis, and so the TCA cycle is turned down, and the catabolic pathway of glycolysis is turned down, and the anabolic pathways of gluconeogenesis and fatty acid synthesis are turned up. We have enough energy, we have enough building blocks. It's time to store some of this fuel for later. And so we've looked at a slowdown of isocitrate dehydrogenase in times of high energy charge in the cell. And we've seen how that inhibits the entry of carbon into the cycle by way of acetyl-CoA. But there are other ways for carbon to get into the TCA cycle. Please do not memorize all of these entry points yet. There will come a time when you're going to have to know this, but we'll save that one for later. What you do have to know is that carbon can enter the TCA cycle at several points. Carbon from amino acids and fatty acids can enter by way of pyruvate, oxaloacetate, fumarate, succinyl-CoA, and alpha-ketoglutarate. If the TCA cycle itself is slowing down, malate can go out into the cytosol to make glucose in gluconeogenesis. But some of this malate is going to continue on through the TCA cycle in the backwards direction until it hits an enzyme that can't go backwards. Now, which one is it going to hit first? Do you know which one? I'll give you a minute. It's going to hit alpha-ketoglutarate dehydrogenase complex, and that one can't go backwards. So what's going to build up? Succinyl-CoA. Do you remember what succinyl-CoA does? It inhibits alpha-ketoglutarate dehydrogenase complex. It also inhibits citrate synthase, and so it further shuts down the cycle. It says we have enough energy, we have enough building blocks, let's turn this down, let's make things that we can store. That either means glycogen or fats. When the cell needs energy in times of low energy charge, this is low concentrations of NADH compared to NAD+, and low concentrations of ATP compared to ADP or AMP, the cell is going to use the TCA cycle to generate energy from fuels. And so the fuel that comes into the TCA cycle, whether it comes in through glycolysis or through the breakdown of amino acids or odd-number fatty acids, will be used to generate energy. In this way, the TCA cycle can be both catabolic and anabolic. And so a cycle or pathway that can do both is called amphibolic. This marks the end of Lecture 20. Have a great day.